from University of Pittsburgh.
Alright, good morning everyone. My name is Shahryar Khaliq. I'm a fourth-year dental student at the University of Pittsburgh School of Dental Medicine. My project today is MMP3 contributing to development of periapical lesions and healing response. I worked on this project with Dr. De Silva, Kathleen Deely, and Dr. Vieira. So we know that apical periodontitis is an inflammatory disorder of periodicular tissues caused by persistent microbial infection within the root canal system of the affected teeth. We know that can be due to faulty restorations, it can be due to poor receptor control during the root canal, and so forth. But it has also been proposed that genetic susceptibility may predispose certain individuals to persistent apical periodontitis. Why? Because matrix metalloproteinases, MMPs, are strongly associated with levels of inflammation and play a major role in bone remodeling and resorption. So basically, bacteria or toxic bacterial byproducts stimulate the release of cytokines such as interleukin-1 and tumor necrosis factor alpha, prostaglandins, and hydrolytic enzymes. These cytokines induce expression of MMPs by proteolytic cleavage of substrates. Once activated, MMPs promote subsequent activation of pro-metastatic factors, which ultimately leads to activation of the Wrangle pathway. This pathway is shown to activate osteoclasts and be critical to bone metabolism. So basically, high levels of MMP gene expression has been linked to high levels of inflammation and carries pulp-like periapical tissue destruction. Expression of MMPs, like I said before, is frequently induced by cytokines associated with bone resorption. We're also looking at TIMPs, tissue inhibitors of metalloproteinases, as these inhibit MMPs. So our study goal is to establish whether polymorphisms in MMP genes and their regulators may lead to an individual's increased susceptibility to persistent apical periodontitis and pulpal tissue destruction in response to deep caries lesions. So we selected 1,600 radiographic records from the University of Pittsburgh School of Dental Medicine's Dental Registry and Data Repository, and we screened these subjects for deep caries lesions that reached the dentin. We also checked for the presence or absence of periapical lesions of greater than 3 millimeter diameter. Based on this criteria, we selected 268 cases. All participants were given an informed consent to sign, and they all provided a saliva sample as a source of their genomic DNA. Out of these, we had 268 subjects who were selected and sorted into two groups. The first group, 158 cases, had deep caries lesions in dentin, but no periapical lesions. The second group had periapical lesions of 3 millimeters or greater diameter, in addition to deep caries lesions. So as you can see in the X-ray on the left, a patient has a deep caries lesion, but no periapical pathology, or no periapical lesion, and the individual X-ray on the right side has periapical lesions in addition to the deep caries lesion. So all individuals selected for the study had thermal and electric vitality tests performed previously. So cases presenting with apical periodontitis had necrotic pulps, and cases presenting with no apical periodontitis were diagnosed as vital pulps. We selected 16 single nucleotide polymorphism markers in the MMP2, 3, 9, 13, and 14 genes, as well as TAMP2. We selected these for genotyping, and results were generated by endpoint analysis in a real-time PCR machine. The SNPs were selected based on previous published reports of their function, and also due to their location or functions within genes of interest, such as location in promoters, exons, or near the exon-intron boundaries. Controls and cases were compared, and allele and genotype frequencies were calculated using the peeling program. We also made sure that heart-rate equilibrium was tested for in the control group to detect genotyping errors for this study. So looking at our results, evidence indicated an association MMP marker genotype RS639752 in individuals with periapical lesions. So the first column on the left side will show you the genotype, and the two columns on the very right side will show you the percentage of differences between cases and controls for these genotypes. The second significant marker is RS679620 in the same MMP gene. And once again, you can see clearly the differences between cases and controls with a p-value of 0.004. So what is MMP? MMP produces stromatolisin, which is involved in the physiological breakdown of extracellular matrix in normal physiological processes, such as embryonic development, reproduction, and tissue remodeling. And the MMP3 variant, RS679620, is a missense mutation, shown to alter wild-type gene function as it substitutes a lysine for glutamine in the final MMP3 protein product. So we believe that such a variant could serve as a marker for faster caries progression into dentin and periapical pathology, due to the strong association of this variant with individuals who develop periapical pathology. Additional analyses were performed for altered transmission for MMP2 marker haplotypes. So we had 3 different genes, MMP2, 3, and 9, that we were testing more than one SNP for. So because of that, these are going to be on the same chromosome in the same gene. So we wanted to check if there was altered transmission together in individuals with or without caries, with or without periapical lesions. So the only significant result was for the MMP2 gene. So like we said, haplotypes refers to SNPs on a single chromosome of the chromosome pair that are statistically associated or inherited together. So looking at our table over here, the first 2, starting from the left side, the first 2 SNPs are not significantly associated or transmitted together. Neither are the second 2. The third and fourth are significantly associated, aka inherited together. So are the fourth and fifth, and the fifth and sixth are not. So basically, the third, fourth, and fifth SNP, as I'm just going to label them, 3, 4, and 5 over here, are significantly associated or inherited together in individuals with periapical lesions. So what is MMP2? It produces gelatinase A, which is a membrane-bound protein that functions in extracellular matrix turnover by cleaving collagen types 4, 5, 7, and 9, and also gelatin. In conclusion, there's a significant association between MMP2 and MMP3 genotypes and the presence of large periapical lesions in addition to deep caries lesions. And these MMPs may contribute to periapical susceptibility in these individuals by varying levels of inflammation, bone remodeling, tissue regeneration, and response to deep caries lesions. Like we said before, MMP3 produces stromatolisin, which is involved in the physiologic breakdown of extracellular matrix. MMP3 genetic variant RS679620 is the one we mentioned second. This one has a wild-type mutation that leads to differences in protein function. MMP2 produces gelatinase A, which is a membrane-bound protein, like we said before, that functions in extracellular matrix turnover. So why are these results significant? Because we're hoping that these results may help predict susceptibility to developing periapical lesions and expected healing response in hosts, potentially helping us predict a chance of successful endodontic outcome, or whether we should look at other treatment modalities. We're also hoping that patients with higher risk to pulpoid and periapical pathology can be identified in the future using emerging treatment approaches such as salivary diagnostics for genotyping or in order to personalize treatment. Also, results may help customize treatment approaches to patients with higher susceptibility. We can use materials that are more bactericidal, or we can use earlier intervention or greater frequency of post-op follow-up to ensure the treatment is successful. Also, additional studies should be performed in other populations to focus on genes identified, MMP2 and MMP3, to further investigate their contribution to variants in host tissue destruction and periapical lesion formation. Before I open the floor up to questions, I want to thank the subjects who enthusiastically participated in the study. I want to mention that the data for the project was provided by the Dental Registry and Data Repository, which is supported by the University of Pittsburgh and NIH grant number listed over here. I will also want to mention that I was supported by the CTSI Startup Program Pre-Doctoral Award through the Clinical and Translational Science Institute and the Institute of Clinical Research Education at the University of Pittsburgh, NIH grant listed over here. And I also want to mention that this project was also supported by my mentor, Dr. Vieira, who was supported by the NIH grant number listed over here. And I also want to thank the Hinman Research Symposium and their sponsors for their generosity. Questions? Yes.
Have you, or do you plan to confirm your data with regard to taking samples from those patients that have MMP2 or 3 polymorphisms to see if they indeed produce more proteolytic activity or MMP2 and 3 activity using zymograms, for example?
So this project was performed last year, and I believe people in the same lab that I'm working in are currently working on that. So we all took a part of the project, and I believe we're actually already working on that. Yes?
Did you notice a difference in the demographics of people with the mutation? For example, did they have, like the MMP mutation patients, did they have earlier breakdown than?
I actually did perform that in a different study, and we did not see a difference in age or gender in these patients.